Can you hear me? Great. So, Kia, thank you so much. It's really a pleasure to visit the Tsai Institute and especially to be Kia's guest. There were two interesting things about this invitation. And I must say, I paid attention to the small print just a couple of days ago, and I saw that I was supposed to be inspiring. Now, that's a little bit of pressure on the speaker. So I'll give you the self-serving advice. The best way to enjoy something is to have low expectations. So the second thing that was interesting was I was given the marching orders to give you a synopsis of my career in 5 minutes. So I will do that, and it will be, I hope, much more modest than Kia's wonderful introduction. But I had to prepare a slide for that because I tend to forget some details sometime. So let me show you the way that I see how things evolved. My career, I would say, started with a thought experiment. I was a junior at Harvard College and taking a laboratory course on psychophysiology. So we were using a glorified polygraph to do certain cognitive tests. And one weekend, I took my then girlfriend up to the laboratory in William James Hall, and I hooked her up to, heart rate, blood pressure, galvanic skin response, and I asked her to think of the most exciting thing she could imagine. And I can tell you the pens flew off the chart. Now, I respected her privacy. I never asked her what she was thinking about. But that got me to think about the mind-body issue. Now, how is it possible for a thought that doesn't weigh much or is not known where it's located to have such a powerful influence on the autonomic system? But that didn't take me very far because the question was nebulous until I attended a lecture by Norman Geschwind when I was a second-year Harvard Medical School student. And then I realized that the mind-body question doesn't need to be nebulous, that it could be parsed into specific networks with a known anatomy, each dealing with a certain domain of human cognition. So that made me decide to become a behavioral neurologist for my clinical identity and do neuroanatomy for my science. So there, as Kia mentioned, I was able to come up with a method for tracing through axonal transport cortical projections in the rhesus monkey brain. And as part of that, I discovered the origin of cholinergic pathways in the human brain. So this last discovery was very lucrative because those were the days when Alzheimer's disease was thought to be a disease of decreased cholinergic innervation. So I got showered with grants. I got the Javits Award for my NINDS. I got the McKnight Director's Award, and I got an SF granting, and so on. Now, while these things were happening, then there was this revolution in the neuroscience world with functional imaging. And as Kia mentioned, I was part of the beginning of that, as was Kia. And I owe everything I know about functional imaging to Kia. And as part of that, we started this beautiful, one of the best episodes of my career, beautiful transcontinental collaboration on the network for spatial attention. Now, while, again, these things were going, I was also making a salary. I had to live. So this was the clinical part. I saw patients in the clinic. I established something called the Behavioral Neurology Unit at Harvard, which was one of the first multi-departmental entities combining clinical work with science, dealing exclusively with human cognition. And during that, I came up with a new syndrome called primary progressive aphasia, which sometimes is known as Mesulam's disease. And that acronym bothered some of my colleagues. They would call me and say, "What's wrong with you? What kind of disorder do you have? We're so sorry. You're sick." And I would have to explain that that wasn't exactly the meaning of the acronym. And then I had an offer I couldn't turn down from Northwestern. I came to Northwestern, started a much larger type of behavioral neurology unit, and established the brain bank, which then got me interested in neuropathology and became my research focus, which continues now, as I will show you. So I really consider myself an opportunistic forager of brain function. I'm not loyal to any method. I'm not loyal to any question. I just sniff around for methods I understand and then find a question that fits the method and try to put a little piece on a jigsaw puzzle that's mostly still open. That's basically my 5, maybe 6-minute summary of my career. So now let me start the real story of the day. And I will take you back to the days when the core belief in behavioral neurology was that you could surmise the function of an area in the brain by studying the dysfunction that was caused by destruction. And one of the greatest practitioners of that approach was Carl Kleist, which came up with this map of cortical function by studying soldiers who had received shrapnel wounds in World War I. And as you can see, there is a place for every cortical function, and every cortical function has a place, except one. You see this area is empty. This is area 38 of Brodmann. Now you will remember in antique maps, the South and North Pole are indicated as terra incognita. So this was the terra incognita, not just of Kleist, but really of neuroscience at that time. And this is the part of the brain that I'm going to be emphasizing today. And to give you context, I will start with a few words about primary progressive aphasia. So this is a neurodegenerative disease, and for those of you who are not familiar, let me define neurodegenerative. By that we mean a disease that arises from the inside of the brain for reasons that are not known, and that destroys certain groups of neurons for reasons that are not known. The best understood or the most known neurodegenerative disease is Alzheimer's disease. But as I will show you, there are others. So this, including primary progressive aphasia, a syndrome that can be diagnosed when 3 particular criteria are simultaneously present. So first, there should be an acquired impairment of word finding, word comprehension, or sentence construction. That's what we mean by an aphasia. Secondly, the language disorder should initially arise as the principal deficit without equivalent impairments in behavior or memory. This makes the aphasia primary. And thirdly, the cause should be neurodegenerative, which makes the aphasia progressive. There are 3 basic subtypes of primary progressive aphasia: agrammatic, logopenic, and semantic. The agrammatic subtype is characterized by impaired syntax and morphology and low fluency in speech with impoverished sentence construction. So if you give a patient with this subtype these 2 pictures and ask the patient to point to the boy kissing the girl or the boy kissed by the girl, they will make errors, because even though they understand the substantives, they will not decode the effect of syntax on sentence meaning. And also they will make similar errors of syntax when they speak, and this will be in clipped, impoverished sentences. The principal neurodegeneration, marked in the form of atrophy and shown here as yellow and red patches in this subtype, occurs in the posterior parts of the frontal lobe, including Broca's area, which is indicated with the B right here, and the underlying disease is generally a tauopathy. In the logopenic subtype, the main problems are in faulty word retrieval and poor auditory working memory. The principal neurodegeneration is in the temporoparietal junction, including Wernicke's area, shown here with a W, and the underlying cause is a highly atypical form of Alzheimer's disease. In the semantic subtype, the main problem is in word comprehension. The principal neurodegeneration is in the anterior temporal lobe, and the underlying neuropathology is an abnormality in a protein known as TDP43. Now, as you can imagine, both the logopenic and semantic subtypes have object naming impairments, but with different mechanisms. The naming impairment in logopenic patients is one way, which means if you show them the object, they cannot name it, but if you give them the noun, they can point to the correct object. So that means that the problem is not in object recognition or in the comprehension of the word that denotes the object, but in linking the two. This is one way. In the semantic subtype, it's different. The patient cannot name the object, and if you give them the noun, miss points, because the meaning of the noun that denotes the object cannot be understood. Now, if you remember, classic behavioral neurology assumes that language is coordinated by a left hemisphere network that is centered around two areas. Broca's area in the frontal lobe that is critical for production of language, and Wernicke's area in the temporal lobe that is critical for comprehension. Now, work in semantic PPA shows that this model is incomplete and that the anterior temporal lobe needs to be inserted into the language network as a third critical node important for word comprehension. Now, this evolution of our understanding of the language network has caused considerable distress in some fields of neurolinguistics. So I should point out that we continue to have great respect for Wernicke's area, and we understand it is very important for word retrieval and auditory working memory. But it's also important to realize that Wernicke's area now has to relinquish the importance that traditional neurology gave it in word comprehension and transfer that responsibility to the anterior temporal lobe in the left side. So now let me show you a little video of what a patient looks like with an anterior temporal lobe atrophy as part of semantic PPA, and we'll see what you think. So this is a coronal MR scan of that patient. It's cut so that this is the top of your head. The ears are here and here. And now as you look at this picture, you may think I'm confusing my right with my left, but that's really not the case. The radiological convention is to have the left side of the brain on the right and vice versa. Why that is beyond me. I think that radiologists probably created this so that only radiologists can read these kinds of pictures. But that's the way it is. Here is the anterior temporal lobe, and I think you can see here that it's much fatter and plumper on the right side of the brain than the left, indicating there has been destruction of this as part of this disease, and the rest of the brain really looks pretty good. So I put these objects in front of the patient, and you can see, I think, that these objects belong to 4 different semantic classes. So there are animals, there are fruits and vegetables, there are tools, and there are other edibles. The patient was unable to name any of these, so now I'm going to give the patient the noun and ask her to point. And as I mentioned to you before, because this is a two-way problem, the patient is going to mispoint. But I want you to pay attention to the pattern of mispointing. And as you will see, when the patient misnames to a noun I give, the mispointing is always, almost always, to another member of the same semantic class. You see a bagel, just touch it, point to it.
Okay, so a bagel, is that a bagel?
Okay. And do you see a rooster?
A rooster. Is that him?
Peppers.
Peppers? Ooh, that's a good question. Peppers. That might be that.
Okay. And do you see a zebra?
A zebra. That is an animal. And a zebra might be that one.
And do you see a waffle?
A waffle?
Okay. And how about an artichoke?
Artichoke? An artichoke. Would that be it?
How about a lime? Lime.
Lime? Lemon. Lime. Yes.
Which of these are dangerous? Would you run away from?
That would be that one. And these two?
So it seems as if the mapping of meaning has been distorted, and the boundaries of meaning that separate words that denote members of the same semantic category are blurred, and the boundary of meaning separating words that denote objects in different semantic categories is preserved. So the person is understanding words generically, but not specifically. Now, you could argue to say, no, this is the wrong interpretation. The problem really is in object recognition. But you can counter that argument because in my last question, when I said which are dangerous, she was able to point to the 3 of the 4 appropriate items. So the problem here is in understanding the word that denotes the object. Now, as the disease progresses, then even the boundaries across semantic categories is impaired, and at that time, as you can imagine, word definition is severely impaired and doesn't respect boundaries of semantic categories. So let me show you what this patient does 2 years later. And what is an artichoke?
An artichoke? An artichoke? Not sure. Is that something that bites you?
And what is broccoli?
Broccoli? Broccoli, is that a bird?
And how about an eggplant?
Eggplant? An eggplant. That's another critter that's growing. An eggplant.
Okay. And how about bagels? What's a bagel?
A bagel. A bagel is a critter. Grows in trees and thickets.
Okay, thank you. Okay, so this is 2 years later, as you see the progression, and using this kind of longitudinal data, we were able to separate the stages of semantic PPA into 3 steps. First, the severe anomia with the atrophy confined to the absolute tip of the anterior temporal lobe, the terra incognita of Kleist. Then in the intra-category blurring stage, which I showed you in the first video, there's a little bit of progression more posteriorly in the temporal lobe. And then at the stage of inter-category blurring that you saw in the third one, going a little bit further back, but still the center of gravity is in the tip of the temporal lobe on the left side, with this obvious asymmetry throughout the stages. So you can see, at least in the first 2 stages, that the Wernicke's area, which is here, is completely intact, again supporting the conclusion that word comprehension is not Wernicke, not in the Wernicke portfolio any longer. Now, there are also patients where the anterior temporal lobe atrophy is in the right side of the brain. And here is such a patient. This is the MR scan, and you see the right side of the anterior temporal lobe is atrophied, whereas the left side looks quite normal, and so does the rest of the brain. And now let's see what this patient does. And by the way, this patient also will show a 2-way naming deficit. What's this?
I don't know.
And what's this?
Don't know.
What's this? And what's this?
Don't know. The names come to me.
And is there an alligator?
Can't, no.
And you see rhinoceros on the tail.
Couldn't tell you.
How about a lion?
No.
Are these objects that you see here as dangerous that you would run away from?
Gee. I don't know.
And what's an elephant?
An elephant is an animal that you find at the zoo.
And how about a lion?
A lion. Okay. They're ferocious.
And how about a cow?
Cow.
Tell me about a cow. What's a cow like?
They're owl.
What do they do?
They're on farms.
And alligator? What's an alligator?
It's a animal that lives in the water.
And so are they dangerous?
Yes.
So what you see here is that, as opposed to the other patient, her word comprehension is perfect, but she cannot name or recognize objects, and this is known as a visual associative agnosia. So the problem here is in nonverbal recognition of the object with the right anterior temporal lobe atrophy. So here really we have the Aristotelian brain. Now, you may know or remember that Aristotle divided semantics into 2, the symbola and the pragmata. So the symbola brain is on the left side, the pragmata brain is on the right side, and obviously they interact with each other to allow us to do our daily living activities. And what is surprising is that both this patient and the other were able to maintain their daily living activities. The first patient, for example, at the time that I showed you the second video was still driving and dealing with finances, socializing very well. She was actually a painter and brought one of her paintings, which hangs out on the wall of my offices. And so if you were in a cocktail party, you wouldn't even know that there's a problem unless you stared at her and say, "What's a pumpkin?" And then you would find out that things are really not doing well. So the question you might ask now is that what kind of disease is it that has this kind of anatomical knowledge to go for the anterior temporal lobe? And here I need to give you a primer on neuropathology. There are really three major neurodegenerative diseases that we see in the clinic. There are the plaques and tangles of Alzheimer's disease. There is the synucleinopathy of Parkinson's disease and Lewy body dementia. And then there are the frontotemporal lobar degenerations, which can be divided into tauopathies and TDP-43 proteinopathies. I'll have nothing to say about tauopathy. Now, TDP-43 is a normally occurring nuclear protein that plays a very important role in RNA trafficking, transcription, differential splicing. And in TDP-43 proteinopathies, TDP-43 is displaced from the nucleus. It's truncated, it's hyperphosphorylated, and it deposits in the form of insoluble clumps in different parts of the neuron. And depending on the shape of these deposits or inclusions and their distribution, TDP-43 proteinopathy is classified into types A, B, and C. And this is where the story becomes interesting. That A, B, and C both have the same abnormal protein that can be identified by the same antibody, and yet they behave very differently. Type A and B have highly variable anatomy of neurodegeneration. Type C always, with no exception, hits the anterior temporal lobe. And in neuropathology, whenever you say no exception, that's really exception. And this is what this disease shows. 30% of A and B are genetic. TDPC is never genetic. A and B are rapidly progressive. C is the most slowly progressing neurodegenerative disease we see in the clinic. Cortical inclusions are cytoplasmic and nuclear in A and B, and they are in terms of dystrophic neurites in C. Let me show you what this looks like. So all three panels here are stained with the same antibody to TDP-43, and the section is counterstained with crescent violet. So this, and by the way, the TDP-43 deposits are these dark brown profiles. So in type A and B, the inclusions are within nuclei, or in the cytoplasm, or in the form of short neurites. Very different in type C. And here you see the deposits look a lot angrier. They are in the form of dystrophic neurites. Some have kind of Y-shaped, sling-like bifurcation. Some others look like these ancient clubs. There are some that looks like drumsticks and so on. And the concentration is at the junction between layer 1 and layer 2 right here. But there is also bilaminar distribution, as you see here. Here is the concentration in layer 2, which you see here in higher power. And then there's also another line of dystrophic neurites in layers 5 and 6. Now, there was a time when these neurites were thought to be in the axons. In other words, the conclusion was that TDPC hit the anterior temporal lobe, especially in the axons. We now know that this is not the case. This is work from a very talented graduate student in my laboratory, Allegra Calls, And here we're looking at a much higher magnification at these dystrophic neurites. Here on the left, what you see is immunohistofluorescence with an antibody to TDP43. So it has fluorescence that's yellow in color. Here she took the same section and also stained it with an antibody to dendrites, which you see here in red. And what I think you will be able to see, well, maybe not in this light, that the dystrophic neurite is inside a dendrite. So the conclusion is that TDP-C hits initially the dendritic component of the neuron. And that's really critical because the dendrite is the intelligence gathering part of the neuron, as you see in these pink filaments that are covered by these synapses shown in as yellow spots. And this is the part of the dendrite that's the most important in information integration. And this dendritic location is not seen in types A and B. So here then we have the puzzle now. Type C goes to the anterior temporal lobe, gets the dendrites, and yet A and B that have the same protein abnormality do no such thing. So the answer to this puzzle came in a paper we published in Nature last year, showing that what we thought was an exclusively TDP43 deposit and inclusion in TDP-C actually is a heterodimer, where TDP-C is TDP43 is bound irreversibly to another protein known as annexin A11. And there is no other neurodegenerative disease where the precipitate is a heterodimer. Annexin A11 and TDP43 meet each other somewhere between RNA grains and lysosomes. They fall in love, never to be parted again. They deposit always together. So we have a definitive molecular explanation of why type C behaves differently from A and B, because A and B do not show this kind of consistent pattern. So the next question here now, okay, we know that they are different, but why does TDP-C choose the anterior temporal lobe and the dendrites? Obviously, the answer couldn't be the normal distribution of TDP43 because that's true for A and B as well as C. But how about the distribution of annexin A11? And here is another interesting twist in the story. So now we're looking at immunohistofluorescence with normal annexin A11 in the normal brain. So all of us in this room, I hope, have this kind of normal annexin A11 distribution. And you can see that there is kind of this bilaminar pattern on the left in layer 2 and layer 5-6, which is also the bilaminar pattern that we see in the deposits of TDP-C. And even more interestingly, here we're looking at layer 2, and we have preliminary evidence that layer 2 has the highest concentration of annexin A11, especially in the temporal pole. And also, even more interesting, that annexin A11 normally occurs in the dendrites, which are right here, and TDP43 doesn't occur in the dendrites. It's in the nucleus. And then another interesting twist that we found that 4 common variants of ANXA11 are associated with TDP-C, but not with TDP-A and B. And another interesting twist, the thalamus never contains TDP-C precipitates and has no annexin A11. So our hypothesis is now that annexin A11 is acting like a Pied Piper, attracting like a magnet the deposition sites and deposition location for TDP-C, and that's why C looks so different for A and B. So now we have definitive proof that C is different from A and B, and kind of a conjecture that why it hits the anterior temporal lobe and the dendrites. And the next question about why does such a small area of atrophy in the anterior temporal lobe cause such dramatic word comprehension impairment? And here we're in a little bit of thinner ice. I hope nobody in this room knows anything about entropy or chaos theory, because I will be using those two constructs to tell you, give you sort of a simplistic approach of what I think is going on. And first, let me just point out that when we name or understand words that are familiar, the process is so automatic that we don't even think that any neuronal activity is necessary. But when things are less familiar, as long as they are within your potential knowledge base, things are different. You know, what is the meaning of an ocelot or a cornet? What's the name of this animal, of this tool, of this vegetable or fruit? So at that time, you almost feel that there's an internal kind of activity. There's a little bit of a hesitation. And this is my simple-minded approach of what I think may be going on. So we're starting with an abstract three-dimensional space where the X and Y axes are event incongruity and anticipation aperture of the person's knowledge base. When the two collide, then there is a certain uncertainty that's produced, which is shown by this topographic map. Now, at that point, if uncertainty is high, you're at a stage of high informational entropy, and to use Carl Friston's term, there is increased free energy in that state. Now, neuronal assemblies don't like to be in a disorder with high free energy, high informational entropy. So the assembly as a whole starts to explore other states, perhaps through strange attractors, until the whole assembly settles into a state of low informational entropy by settling into a state that is a least conflict between alternatives. So that particular state of low informational atrophy is what we identify as a solution to our problem, either the correct name, correct recognition, or just simply say, I don't know, or something in between. Let me point out that this reduction of entropy I am talking about is local entropy. That the brain, however, to go from here to here, has to spend metabolic energy, which creates heat that is dissipated to the environment. So global entropy is preserved. It's local informational entropy that is being reduced. And there's reason to believe that the anterior temporal lobe is particularly critical for this kind of process. And here I'm going back to anatomy that is more familiar ground for me, especially based on primate cortical anatomy. And as is well known, the superior temporal gyrus has a multisynaptic pathway for auditory information processing, whereas the inferior temporal lobe and the fusiform gyrus do the same for visual pathways. And words start as objects. They are basically unimodal percepts. And as they get subjected to this multisynaptic chain, at each synapse, through dendritic gating, specific attributes of this information is extracted. So you get from very general to more and more and more specific. And both of these downstream pathways converge in the heteromodal cortex of the anterior temporal lobe, so that unimodal word percepts are linked to the multimodal associations that collectively contain what we call the meaning of the word or the nature of the object. And that's just the beginning. From the heteromodal cortex of the anterior temporal lobe, there are feedback projections that mold the incoming information according to the experience, expectation of the individual, the understanding of the context of the individual. So we each understand words, recognize objects a little differently. Now, TDPC destroys precisely this part of the brain initially, and also the dendrites, which obviously would severely disrupt this process. But TDP43, TDP type C, doesn't completely destroy this area. It's a gradual process. So in the initial stages where you lose a certain percentage of neurons, but not all of them, what you have is that the attractor basin becomes shallow so that you can understand words only in the generic but not specific form, and that, of course, progresses further. This, then, is the conjecture of why the anterior temporal lobe is so important. So we have gone through 3 steps. Why is it that type C is so different from A and B? We have absolutely watertight molecular findings to show this. Number 2, why does TDP-C go to the anterior temporal lobe? And we have sort of a reasonably good hypothesis. And thirdly, why does it interfere with word finding? And this is an ongoing work in progress that we're trying to define it in greater detail, but we have at least the skeleton of theory explanation. That's basically as far as I can go today. As opposed to classic behavioral neurology, which was based on focal penetrating injuries, shrapnel, bullets, or specific strokes, the behavioral neurology of the 21st century is gravitating towards the study of neurodegenerative diseases, and in a way that really allows us to bridge the gap all the way from the molecular aspects of cortical areas and also the computational architecture of what they do. So, thank you.